To address this problem, we used the model, which is shown here. It is based on the assumption that our observation, which is the y, consists of many different contributions, including the neuronal signal, actual neuronal signal from the brain, plus the other confounders denoted by x1 and x2. Then the x1 and x2s will not directly go into the y observation, but it will have some kind of transformation or some kind of dynamics denoted as h1, h2. So these are actually the impulse responses in this model. So h1 in this case will have some memory, and h2 has these memory of few seconds, for example. And when the x1 signal goes into this system, this is basically the convolution of x1 and h1, and that's also added to the convolution of x2 and h2, plus the neuronal signal that's hidden in our y, which will be the Fneer signal that we measure. So in reality, what we picked as a confounder is the end-tidal CO2, because this is a good metric of how much you're breathing and how much CO2 you build up in your brain. Then as where the brain would auto-regulate based on CO2 in terms of brain blood flow, and the X2 as PPGA, which is again its photoplethysmograph amplitude, which is a good indicator of systemic blood volume or systemic perfusion changes. In this case, it will be representing the confounding blood flow in the scalp. But note that this is actually measured on the finger. So again, it is very advantageous to have this H2 because we're not actually directly adding the contribution from the finger blood volume to the system, but we are saying that there's some kind of transformation towards the scalp blood flow that we haven't measured. But then the model will be able to figure this out. And of course, the last row is the Fneer's measurement itself. And as you can see here, constant mean was removed, and there was a band pass filter, reduce unnecessary physiological noise that we're not interested in for this study. And here, the model memory, the M1 and M2 size was up to 30 seconds, which translates that the length of the impulse responses here will be 30 seconds. So let me show you this smaller model before we actually show the effect of the full model. So here we have one input, and this is the neuronal input that they go together, and we are observing the sum of these two. Now, as the title says, since we're using this first order convolution, we're assuming our phenomena is linear. So again, the first order means there's no square terms. And over time, which is denoted by this k, so k will be number of samples, right? So k will be starting from 1 through, say, how many ever samples you have. But over all time, whole time of the measurement, this relationship will not change. So it's time invariant. And it's dynamic modeling because we are convolving the X confounder with some kind of impulse response that has dynamics. And we use this in this case to explain the amount of cerebral CO2, which was again denoted by end-tidal CO2 in our case, that influenced our Fneer's measurement. So these are the signals. And when we set this model, what's unknown is the H and the V, or let's say V, because we measured end-tidal CO2 from the patient and we measured Fneer's measurement. Now what will happen is we'll do our best to find the best fit H that provides the minimal residual in this model. And this should be another sort of a course, but to express this H, there are a lot of different ways. First of all, if the memory size was, say, 30 seconds, you have 30 seconds of unknown values. And you can imagine at each second you have unknown, so there are so many combinations of unknowns to figure out. You can do that, or there's a better way. That is to use Laguerre basis functions. This is proposed by a researcher saying that this H, the impulse response, unknown, would be made of some kind of optimal combinations or summations of their the waveforms here, first order, second order, third order, fourth order, fifth order, denoting that how many zero crossings they have. And you can have different scales on them, and you can add all of them to form this. And to do this, you don't need many parameters. You only need few parameters that will decide how many basis functions you'll use, what's the scale in the x or the time axis, and also the scale of the y-axis. So that's here, these two, and also there are some hidden parameters. And what happened in the model is that we test different combinations of these, sum of all the Laguerre functions. You see this is one of the examples, and see whether there's any impulse response that best fit the input and the output relationship. Now the blue line here is F-near signal, and the magenta line shows how that CO2 signal was fitted into the blue signal using this model. And here there was a model selection criterion, which is in our case was minimum description length, and actually there are more metrics that you can use, but basically the idea is you want to find a model with the optimal accuracy while you're not making the model too complex that would only work on this specific signal. So in theory, you can train this model with many different parameters and different complexity to fit this magenta line perfectly on, almost perfectly on the blue line. But then we will say this model has been overfitted because it will just explain, or it will work well on this case of signal only. But we want to have more a general explanation of the phenomena. So that's why we have this model selection criterion that will penalize the process as I add more parameters while it's looking for the best optimal fit. Now, so you can see that the model, this model, fitted very well for those periods with rapid decrease. You see there's a dip, dip, dip here, which are actually the periods when the subject made a deep breath here. So this is actually the respiration signal, and that caused vasoconstriction, and that was represented, or that was sort of contaminated the F-near signal. And this is of course not brain, but the psi responses. Now the idea is, since we know the contribution of this breathing, we can subtract it from the original signal, which is the blue, and what we get is this orange, which is as a residual, which shows in here, and we can be more confident that this is neuronal component of the F-nears. Now on the full-blown model, so we can do that process with two inputs. So here, as we've done, there's entitled CO2 we talked about, and the contribution of it is about 30% because this is a normalized mean square error. So error of 1.0, sorry, there will be 100% mismatch, and the mean square error of 0 will be perfect match. So this is saying about 31% of the fitting here, and this is about 27% from the PPGA. And you can see that this PPGA fit very well on the rest of the background fluctuation. Now we can subtract all these confounders, right? These are not brain, or so to speak, not brain due to our task. Now the final residual would have minimal confounder. And we've done this process for each of the F-near signal for each patient or subjects. And note that this is the actual impulse response that was found through the modeling, and you can actually appreciate the directionality. As a result, this model-based filtering made the F-nears measurement less confounded. So this is one case before. So this is the 3-bag, one of the difficult tasks. And you see try number 2 and 3 has this unanticipated dip that would hurt our data. And the average change of the oxygenated hemoglobin was also in a weird shape, as there's no clear positive increase on the 2 and 3 bag. And we think that this is probably due to the vasoconstriction you see in the finger blood flow, And now after the modeling, you can see that these dips are much removed. And now we can say the F near signal, or the brain component that we are actually wanting to quantify, is now more independent from the blood flow of the skin and the breathing. This is also seen in the summary plot, where the filtering improved the trend so that it showed a nicer increase towards the difficult task. So we think this model-based filtering worked pretty well.